The overall goal of this protocol is to construct and align a structured illumination microscope suitable for high-speed live cell super-resolution imaging, capable of operating in TIRF and optical sectioning modes with multiple colors.
This method can help answer key questions in the field of cell biology that cannot currently be answered using standard diffraction-limited TIRF microscopy. The main advantage of this technique is its fast time resolution compared to other super-resolution microscopy techniques, which make it well suited to imaging dynamic processes in living cells.
Though this protocol describes the steps for construction of a TIRF-SIM microscope, the setup is flexible and easily modified to implement imaging modalities such as 3D SIM, multifocal SIM, and nonlinear SIM.
To arrange and align the excitation path for a structured illumination microscope, begin by marking the positions of the components on the optical table, as indicated here. After inserting the first dichroic mirror DM4 into the filter cube turret of the microscope frame, insert the second dichroic mirror DM3 into a 1-inch square kinematic mirror mount and position it one focal length away from the condenser lens L5. Next, to accurately define the optical axis for the system, remove the objective lens OB from the turret and instead screw in an alignment tool. Then, use the dichroic mirror DM3 and a temporary alignment mirror positioned at the approximate ultimate location of the SLM to steer a collimated reference beam from laser 1 through the center of the holes in the two alignment discs. Using 3 mirrors and dichroic mirror DM2 as depicted here,